Sorry.
But I
Oh.
I feel like sometimes like everything's just a little too cold.
Well, I need to put the
Like,
mic-
oh,
Yeah. To be fair though, I grabbed
Yeah,
it for
go ahead.
breakfast this morning and then I felt too awkward to eat it in class during 372. It's just too quiet for me. So I held it in my lap the whole time and then I ate it in the next class. So she was a little warm by then.
I genuinely do not.
That's going on
All
YouTube,
my
by the
eyes
way.
recorded.
That's going on YouTube.
Well,
That's going on YouTube. It's going on YouTube. That's also going on YouTube.
So this is why he shouldn't bring the mic.
All right, hi everyone.
Hi, John.
Topic 3 review. Nice. The hardest topic by like a lot. Yeah.
Oh, yeah.
Yeah, but I'm like no expert in immunology, so I'm trying my best here. We're trying our best here. This topic's hard. So just come on. Just a little outline. God, I hate this projector. This projector
This
sucks.
might be a little bit of content though,
Mm-hmm.
just a bit,
This projector sucks.
but
So, yeah, so that's the outline. I'm not gonna read step by step, but we'll go through all of that for the review, yeah. Fun times. So, this is just a little, God, I actually despise this projector, but this is just, can we turn the light on? Yeah, maybe we'll do something.
Oh.
Okay, that helps a little
That's
bit.
actually pretty.
That helps a little bit, that helps a little bit.
Five.
No, there's, no, Jen, there's a chart, there's a, there's a.
Oh.
Orange strip right there. Okay. So, this is just a chart, it's like a time frame of, the two different immune systems that we have, so the innate immune system and the adaptive immune system, and how they, respond to pathogens and to antigens. And so on the darker blue, it's the adaptive immune system. As you can see right here on the left side, there's a little bit more of a lag phase. Because the immune system that starts the response upon pathogen invasion and to prevent pathogen invasion is the innate immune system, and that's the lighter blue. And once it deems itself not strong enough to deal with the invading pathogen, if necessary, then it calls upon the adaptive immune system. So we're gonna do innate. Okay.
so your innate immunity, this is a prehistoric system and it's found in like every single multicellular organism. And this is generally like the first line of defense against pathogens, and it's characterized by being pretty completely non-specific. So it's just kind of it will attack any sort of foreign thing that it senses is in your body. And you know, this is super important because your body, believe it or not, takes in a lot of things that shouldn't be there, like single bacterium, on non-living like things, like even like, I don't know, like a splinter or something, you know, these things need to be somehow, you know, dealt with and expelled or whatever, destroyed and broken down. And this happens really quickly after invasion. And the mechanism is generally conserved for all of them. So this, like I said, like non-specific, it will treat every single, most like things that it senses in the body that isn't the body, the same. And you know, this also includes like the skin being a physical barrier in protection, along with like different immune cells. And you don't have to know all these immune cells, but it's good to generally like know which ones are in which group. So like neutrophils, natural killer cells, mast cells, dendritic cells, eosinophils, basophils, which we just learned recently why they're named so.
Dude, their names are just, they're not even picture sick. So right there on the top right, it's a picture that I took last Tuesday, like 2 days ago. And we were talking to Dr. Spracklin in histology class, and he was talking about the different granulocytes. The eosinophil, neutrophils, and basophils are all granulocytes. And so there's this type of staining that goes along in histology, which is the exact staining that's used in that picture. It's called hemotoxin and eosin. Hemotoxin is a basic dye, and so things that are acidic in nature will tend to be stained by it, and that stains dark purple like the nucleus. And there's this other stain that's used at the same time. It's called eosin, and eosin has a acidic profile, and so things that are neutral or basic will tend to bind to it and tend to be stained by it. And so that cell that you see right there is it's a eosinophil, and that you can say that it's a eosinophil, Jesus Christ, because it's stained with eosin.
This doesn't matter. We just think it's cool.
It's like super sick.
You guys
Like-
don't need to know.
Like a
dude.
basophil is called a basophil because it's attracted to basic dyes. And it shines dark purple when you see it under a microscope. And a neutrophil, you'll see dark and you'll see bright pink, and it's basic because of that. Alright, and it's neutral because of that, sorry. Yeah, good.
Anyways.
Yeah.
Do you want to do adaptive or?
Yeah, I'll do adaptive.
Alright.
Alright, adaptive immune system. So these are different. So as the name implies, you'll have adaptations to prior invasions by other pathogens. The only two components of the adaptive immune system, like legally speaking, are the B and T lymphocytes. Though you have cells that are supportive of the B and T lymphocytes for their functionality. So you have your NK cells and you have your dendritic cells, you have your antigen presenting cells, and you have your memory cells. And those are all supportive cells to the adaptive immune system, but they themselves aren't part of the immune system. It's just the B and T cells. And so you have recognition and targeting of identified exogenous particles, and we hope they're all exogenous. And you have hypervariability in the ABD of antibodies, that is the antigen binding domains. And we will talk a little bit more about how that process happens. It's very complicated. And so the fact that this region is hypervariable means that you have the ability of binding to a variety of different unknown substances that will have the same sequence. And that just happens by chance. I think the number is close to like 10 to the 3 times 10 to the 23rd possible recombinations that can happen. So like if you put that in a calculator, it won't give you the number because it's just an error. It's a stupid high number, basically is what I'm trying to say. Huh?
For antibodies?
Yeah.
1 quintillion.
It's like stupid. It's like a stupid number. And so yeah, so you have memory cells, and then you have the immediate plus stronger response upon second exposure. Basically what that means is if you've gotten the flu before, your body will have adaptive cells that have attacked those cells before. And so upon reinvasion, upon the pathogen being able to re-enter your body, they're already prepared for the invasion to happen because of the higher number of antibodies that have been produced. Any questions so far? No? You wanna do this?
Sure. Antigens. So these are basically what is targeted by the adaptive immune system using antibodies. AIS, I've never seen anybody call it adaptive immune system. AIS, that's adaptive immune system. These can be sourced endogenously and or it can be like and or exogenously. Let's see, these have a variety of different identifiable sites. So the epitopes are where the antibodies bind, and they can vary in terms of their structure. And like because they're very in structure, it can vary in like what kind of antibody can actually bind to it. So you can have the immune system produce multiple antibodies with different binding sites that will bind the same antigen, just at different epitopes. Right, and that kind of helps that like cohesive response to an antigen. And yeah, like they can be literally anything. It doesn't have to be a living. Does it have to be living?
It could be a glycoprotein, it could be a lipoprotein, it could be just a glyceride, it could be like a
Yeah.
saccharide, it could be anything.
Anything that's foreign, basically, we consider antigens.
What's the endogenously?
Endogenously means it's produced from the body,
Okay.
like within the body, and exogenously means it's from outside the body.
Thank you.
Would the endogenous ones be autoantigens?
We'll get there.
So,
No spoilers.
Yeah.
Maybe like top end.
Want me to do this one? Do you want to do this one?
I can start it. So antibodies, these are kind of like the focus of this topic, right? This is where the BDJ comes in, but these are produced by the adaptive immune system, specifically from the B cells in response to a specific antigen. So basically the structure of this consists of this, like, you've probably seen it before, like this Y-shaped immunoglobulin, and it's basically has 2 identical heavy chains and 2 identical, was it supposed to say identical?
Huh?
Are they two identical heavy and two identical light?
Yeah.
Or did I? Okay,
Yeah.
so they're two identical heavy, two identical light. And those are what come together to form this like Y shape. And you know, it's nicely color coded over there, but you have your light chains in pink, your heavy chains in blue.
Can I just say something right before that?
Yeah.
So each of these chains will have the two regions. So you have a constant and a variable region. But it's basically a constant region will have its own so one light chain will be connected to a heavy chain. And each of those chains is its own individual protein. So basically one antibody is 4 different proteins together, which forms beautiful quaternary structure. And so
the disulfide bonds, see those?
exactly, so each of the halves of the Y are 2 different proteins. And then each of the proteins within a half of the Y are also their own individual proteins. It's color coded in like different colors right there.
Yeah.
On the bottom, and you can see it easier, but they're 4 proteins.
Basically you end up with 2 antigen binding fragments, so that those are like the ends of the Y, and they're just labeled as Fab. Each of these consists of 3 hypervariable regions, and these regions give rise to the like 3 loops of beta strands, which kind of coordinates how it's gonna bind the antigen. So this like area right here is where those loops exist, and that's like determines the way it's gonna bind an antigen and if it's gonna have good affinity or not.
And the antigen binding domains are pretty small.
Yeah.
They're like 5 to like 15 amino acids long.
Shouldn't the light chains,
Mhm.
the green and the pink,
Mhm.
shouldn't those be not identical?
They're identical proteins, but they're different proteins.
I see.
So there's protein 1, 2, 3, 4. Proteins 1 and 4 are identical, proteins 2 and 3 are identical.
Cool, and then you also have your crystallizable fragment, that FC, is which exists in like the trunk of the Y shape. And this is formed from the constant domains from the heavy chains. And this is where the immune cells bind to, and like recognize like antibodies or like take in antibodies that are bound to antigens. So like all the interactions that take place with the immune cells happen at this trunk. So like antigens bind here at the ends of the Y, and then your immune cells interact with it here.
That's also where they're originally bound to the B cells.
Yeah, and any sort of variation in this constant region, like between antibodies, kind of can define the class of antibody that exists. And there's many different classes that exist. I don't think you need to really know any of them, but just recognize that like there are like groups of antibodies that exist, like certain types that complete certain functions. I don't know what that gene is. I think John put that.
It's the bottommost part of the antibody that actually touches the cell membrane. It's expressed, the protein is the FC protein, and it's expressed by that gene, but it's too specific, you don't need to like.
Yeah.
I did it because somebody asked me about it.
You can easally go down a rabbit hole in this content. They're just like
Yeah.
know as much as you need to, or else you're gonna be stuck like.
But you don't have to like go into like a whole immunology, for, like, it's fine.
Cause I'm probably like immunologist or something like.
I mean, I respect immunologists greatly. Dude, it's so much to know.
We're not doing tangents now.
Yeah, it's just, it's connected in so many ways and you need to know so much, but like for our purposes, just know enough to understand the papers and you should, well. No, I don't think anybody really understands the papers, but know enough to like have a good grasp on the general idea of what's going on. You don't need to know like this protein specifically in this unless we tell you like you need to know this protein. Okay, next one. VDJ, all right. I'm probably gonna miss some things, but we'll get into the basic process of how this works. So antibodies, are beneficial to us in binding antigens because they have so much variability. And that variability comes from like mixing and matching different genes and expressing different permutations, I guess, of these things called like the V, D, and J regions that define like what protein structure these antibodies have. So this occurs in the early stages of lymphocytes, specifically in T and B cells. And it involves this protein family or collection of proteins, not so much they're related, but just like a collection of different proteins. In the called the BDJ recombinase. Ones that kind of stand out is the recombination activating genes 1 and 2, RAG, and recombination signal sequences, RSSs.
Hold on. Can
Yeah.
I say something?
Yeah.
This is pretty much alternative splicing on like steroids, like the
Yeah.
strongest possible level of steroids there is. Because you don't need to know specifically the numbers. If you want to know the numbers, like we could talk about it. But there are like more than 20 different V regions, like more than 10 D regions. Like it's like a stupid possibility of like which gets spliced out, which exons remain.
Yeah.
So it's just like by pure chance that the different genes are expressed or not.
And we're also gonna watch a video on this after I talk about it, just like I think the video is nice. John you put the same video I was gonna put.
Yeah.
Yeah,
I
it's a
love
good
that.
video.
I really
So-
love that video.
You know, this graphic I think was okay, but I mainly just put it in there just so you guys can see like where the VDJ and constant regions kind of stand in terms of like the genome.
It gets a little bit more complex and we'll get into it like after too.
Yeah, so basically it starts by RAG1 binding to RSS and surrounding the VDJ coding regions. And then this recombinase is able to, and RAG I think specifically induces the double strand breaks between the gene segments in the RSS. And this kind of forms two things. So you get a hairpin loop on the coding segment, and you'll see this in the video. And then a blunt end on the signal segment. Now I know the blunt end on the signal segment kind of, there's some proteins that are involved that kind of have this like, it takes the blunt end and it like makes like a circular piece of DNA and it's kind of discarded. There is some evidence that it does serve some purpose in like oncology or whatever, but like who cares? We don't need to know that. It's just that part's gone. And basically you end up with these with this hairpin that is allowed to be opened by this protein called Artemis. And what's important about that is in that hairpin, it can be opened either like evenly or off center. And then when it's off center, you have this overhang of nucleotides that kind of adds to variability. And actually in skid mouse models, if you ever heard of those are the mice that are immunodeficient and used in a lot of like research labs. They are made immunodeficient by having a defective Artemis. So because you can't open that hairpin loop, you can't produce any antibodies that are, variable in, binding antigen. So you have this like really immunodeficient living thing.
And we do research on it.
Yeah, it's cool. And then you have this other protein called deoxynucleotidyltransferase, TDT. It adds additional nucleotides.
That thing's crazy.
Very great.
That thing's insane.
Yeah. You want to talk more about it?
Yeah, I can talk about it. So, so what ends up happening is when you have the double strand break that's happening because of the formation of the hairpin and then the singling out of the hairpin, you're left with a double strand break. And you might remember from Bio 25. If you don't, I suggest looking back at Bio 25 notes. But the process that happens when you have a double strand break in DNA is, well, you can have homology directed repair, but that's not the one that we're targeting right now. We'll be using the process of non-homologous end joining. And TDT is insane because typically for non-homologous end joining, what you do is you just join the two ends of the DNA that just got split and you put them together. You lose some nucleotides in the process, but it is what it is. You have for this specifically, you have a double strand break, and then this thing goes in. It's a DNA polymerase, no template. It does not care. It just starts dishing out a bunch of like amino acids or not amino acids, nucleotides with no specific sequence, no specific order, and it's just templateless. So it's like insane. It just goes in there and it's like, oh, we need some here, until it like reaches the end. So it's pretty crazy. And that just allows for like continuous hypervariability to happen, and like the number states for itself like a quintillion. I don't think like it's picturable how big of a number that is.
Yeah.
So we're gonna
Hold on. And then basically after that happens, you get those additional additions. Ends are ligated, and you're left with that like on the bottom, like that region of VDJ that can be transcribed, and you produce this unique antibody. One of 1 quintillion apparently. We can produce up to 1 quintillion unique antibodies, which is a lot, especially considering that like antibodies bind specific antigens, but I'm pretty sure like the affinity that all antibodies bind antigens isn't the same. So you can have some that are like, close enough and they bind. So now you're just opening the door to like way more antigens than specific one-on-one binding.
It's kind of similar to how risk happens in RNA silencing. Sometimes it's not specific enough where like you're having a one-to-one match, but you're still specific enough that it binds it.
Can I go through more? Editing to be more specific.
Yeah, so what happened, more specific, I don't know, but for autoreactivity, and we'll get to it and we'll talk about it, there's this process called, crap, I forget, huh? Yeah, I forget the exact name of it, and I'm gonna look at it on the iPad to like tell you. But if the body recognizes a pattern that is endogenous, it can completely replace the antigen binding domain by a different one. And that happens in the maturation process of like both TCRs and BCRs. But we'll cover it. And now we'll just play the video that's like really cool. See, I told you I needed to fix the audio.
Oh.
Give me 1 second.
Immunoglobulin genes are composed. Immunoglobulin genes.
The microphone is muted. Okay, I'm just gonna have to go over this again, but so RSS spacers are just the recombination signal sequences that flank the gene segments. So each V, each D, each J segment has an RSS, and they can either be 23 or 12 base pairs long. And there's a rule of 1223, so they have to be complementary. If you have two 23s, it won't work as in the H chain, and so you need a complementary gene to come in and have two 12s so that you can actually have the recombination work so that the genes are able to follow each other and be expressed properly. If it has 12-12 or if it has 23-23, it just simply does not work. And so this is a visual of the B cells chains and how the expression happens post DNA splicing or RNA splicing, sorry. And this is a visualization of the T cells. And just another visual of RSS. So here is our comparison between the T cells and the B cells, B lymphocytes, T lymphocytes, whatever you want to call them, they're the same thing. So starting with the T lymphocytes, T cell receptors are not antibodies. They are just called TCRs or T cell receptors. This is important. You have different types of T cells. So you have CD4 plus and CD8 plus T cells. CD4 plus T cells can only bind to MHC1 complexes. MHC is the major histocompatibility complex. It's expressed in humans together with the HLA genes. And so you have a whole bunch of different, like a family of HLA genes that can be like very different. And so MHC1 with 4 plus, MHC2 with 8 plus. And what's very crucial here is that your T cell receptors cannot diffuse away from the T cells, so they're stuck. And the T cells heavily rely on antigen presenting cells and the other memory cells of that aid the adaptive immune system. And that's because your antibodies can't diffuse away, bind to random things and call on for more antibodies and the inflammation response. They have to be presented what they need to bind to. And B lymphocytes, so this is where you do have your antibodies. So B cell receptors are antibodies. And B cells are also involved in antigen presentation for the T cells. But that's a different process that we don't need to cover because it's very complex. And antibodies have a different name, immunoglobulins, that just means the same thing. If you see IG, IgA, IgB, IgE, IgG, it's just different types of different types of antibodies. And immunoglobulins or antibodies are typically released by B cells, and they can be free flowing around plasma, around blood, or around different tissues. And free flowing immunoglobulins are the ones that trigger the different immune responses, so inflammation. But then you also have like chemokine and cytokine storms and a variety of other processes that we don't need to talk about because again, they're very complex. Okay, we'll get that one second.
Yeah, sure. Okay, I actually want to go back for a sec because so just make note that it's not like usually when you have a noticeable immune response, it's not like one antibody binding one thing and then you suddenly have a big response. It's like you have a lot of antibodies binding the antigen, and that antigen is only taken in because like there's a group of antibodies that crowd it, right? The cells generally recognize that like there's like multiple antibodies bound, and that's what allows it to go into like the binding pockets and interact with the immune cells. So just keep that in mind. And then autoantigens. So these are endogenous particles which cause reactivity by maturing B and T cells. These can be proteins, glycoproteins, proteolipids, other molecules or complexes. It's not really specific, so it's a wide range. And then also autoantibodies are, antibodies or TCRs which react to the autoantigens. And then failure in maturation processes leads to the formation of autoreactive antigen binding regions, which we'll kind of talk about in a bit. And like this process together with the autoantibodies and the autoantigens and the failure in correct maturation can lead to autoimmune conditions like type 1 diabetes, other stuff you guys research. Like this is where this stems from. We good? So autoreactivity. So this is the process of B cell maturation in the, so the process of B cell maturation in the bone marrow is called central tolerance. And the process of T cell maturation in the thymus is called thymic selection, which I'll talk about in a second. They are very similar processes, and I think even in the next slide, I mislabeled them as like the same thing, but the general process is the same. Bone marrow and the thymus are both rich in endogenous particles. And this autoreactivity increases the chance for autoreactive antigen binding BCRs and TCRs to be found. Yeah, so I guess I'll talk real quick about the idea of central tolerance and thymic selection. So central tolerance and thymic selection involves removal of any B or T cells that are found to be autoreactive. This takes place in the bone marrow or the thymus, depending on if you're talking about B cells or T cells. Autoantigens of certain cortical epithelial cells are generally used to create like this variety of MHC complexes, and those MHC complexes are essential to how like the lymphocytes work and interact in response to immune or just generate the immune responses that we kind of can visualize. So basically these cells are evaluated first on their binding to these certain peptide MHC complexes. Those unable to bind do not receive the required survival signals, and this is like the positive selection part. So if those immature cells are unable to bind to these MHC complexes in this first stage, then they just stop receiving survival signals. They commit apoptosis.
What pathway is that? Which one? It's like so straightforward. Survival signaling?
Oh, I thought you were.
Oh no, yeah, 3K,
Questions?
Yeah.
Go for it.
These cells unable to bind to antigen, shouldn't those, there's not negative selection?
Wait, I'm sorry.
The ones that can bind.
Yeah,
Yeah, so the ones that bind to the auto.
Is it negative selection?
do I have them flipped?
I think negative selection means like you ignore it.
So
There's also, okay, so typically I'd call center tolerance more for B cells. Because the process that happens in the thymus has a different name. And
thymic selection. You said.
thymic-
I thought they were both pretty similar.
They are pretty similar, but the main difference is the location is like, and what type of cell it targets.
Yeah.
But we will talk about,
Like when you select, like it's Darwin, here,
Sure.
select for something.
Yeah.
You want it.
I'm gonna make sure before I give an answer.
I just thought it was like.
Yeah, this was stupid. That makes sense, but also like.
I trust you.
I see what you're saying, but
I also see what you're saying.
But I know we.
I think to avoid confusion, just don't talk about it. You just say that there's.
It's a
you just
great
say-
tactic.
You just say that the cells that are autoreactive are selected against. Beautiful.
Yeah, that I just wouldn't work. You don't really need to label them. but
Okay.
I will look into if I have those flipped, but I don't think I do.
But yeah, thymic selection is the process.
Unable
for
to
T
bind.
cells, and then central tolerance is the process for B cell.
Okay, yeah. And then so basically they're evaluated based on their binding to cell peptides. Those that do end up binding are selectively pushed into apoptosis. And there's a thing called peripheral tolerance, which calls B cells and T cells outside of the body, not outside of the body, outside of where they mature. And this is kind of, I kind of like to think of it as like spot checking, right? Making sure that these are still working correctly and not binding self things. You have anything else?
Just.
I think I might have it.
It covers on the second last slide. we're good. Okay.
You could talk about whiskeys.
Yeah, so. This paper is a pain to find online for some reason. like, it's like the original paper is like really hard to, I remember over the summer I was struggling to find, I was getting like very mad. But basically the four major postulates from 1957, are that you need direct demonstration of free circulating antibodies active at body temperature. You need the recognition of the specific antigen for this antibody. You need production of antibodies against same antigens experimental animals. And you have experimental animals that demonstrate the same tissue changes as in a human. These are just hallmarks of autoimmune diseases that the dude had, and that he thought was true in 1957. Though he's been proven wrong time and time again, because not all autoimmune conditions necessarily need to display all of these postulates at the same time. There are some that do, but a lot of them don't. Questions? No. And then there's the postulates revisited document from 1993, which, you know, it's approaching, it's pretty old now, like 30 years. But so there are four also, and the first one is that the clinical phenotype can be reproduced through the transfer of autoantibodies and or lymphocytes. That the disease can be reproduced in experimental animal models, so pretty similar to the original. That autoreactive T cells or autoantibodies are identified, which is also pretty similar to the previous one, and that distinctive clinical observations such as HLA association are found. Basically, all that's trying to say is that, and that's where I got the previous statement from, like that I read the original paper in 1983. I also read like a literature review of the paper from last year. That says that, or they say that you can have a lot of the postulates happening at the same time, but it's not required that you have every single one for a disease to be autoimmune. Be it because we can't identify all the postulates at the same time, or be it just because the conditions for the disease are different. Any questions? I feel like out of like everything for this topic, that's like the easiest. Okay. So then I also, there's a question on the problem set that talks about you highlighting 3 different diseases. I think the 3 easiest ones to cover are type 1 diabetes, lupus, and vitiligo. We will not be covering vitiligo or lupus. I wanted to do a spotlight just on type 1 diabetes, just because it's the easiest one for me to talk about. And it's one I'm interested in. If you want to talk to me about it later, we can talk about it. If you want to read my paper on it for your disease mechanism paper, you can also ask me about it. But so type 1 diabetes is an autoimmune condition where beta cells in the pancreatic isles of Langerhans are attacked by autoreactive T cells. And you have different proteins that get targeted for type 1 diabetes. So you have GABA that gets attacked. No, GAD gets attacked. It's not GABA. You also have ZNT8 and sure what's up. Hold on, there's one more. You know, yeah. Hold on. Hold on. Okay, so it attacks GAD, it attacks ZNT8, and then it also attacks insulin. But the attack on insulin isn't the main causation of the disease. It's more because it attacks specific proteins that can't, or that are needed for insulin secretion and like further glucose recognition by the system. And so it causes slow deterioration of insulin production because it's killing these cells. It's not that the person stops producing insulin, but more because the cells that do produce insulin get killed off. So it's a slow decrease in the level of insulin production in the individual, not that it's like ceases at all, or I mean at the same time. It does cease if you don't. Well, I mean it does cease with time because the cells just end up killing more islet cells than the islet cells are reproducing. But this happens because the hormone is produced in the beta cells, and the deterioration of that system leads to a lack of production of insulin at the end. And so like I was just talking about, patients can't sense glucose levels in the blood. And this further leads to high levels of acidity in the blood, diabetic ketoacidosis, and potentially death. You get very dehydrated because the kidneys are trying to make up for the high levels of protons in the blood. And the way that happens is by dehydration. So you have polyuria. And with polyuria, it leads to further dehydration because you're losing lots of water. And so what ends up happening is when you have a lack of glucose or a perceived lack because there's no lack of glucose in the blood, the body thinks it's in starvation and starts producing acetone and other ketone bodies in order to use for ATP production. And so what ends up happening is you have a lot of both resources, and then you just aren't using energy correct and you die. Okay. And then I thought that this was like pretty hard for the topic. I know she only assigned the 4 pages for this paper. And this is, I think, in one of the initial pages of that paper. The paper is super long and like extremely hard to digest. But basically these are just processes that happen due to autoreactive antibodies, but it's not that they're triggering autoimmune responses. They're just preventing the functionality of other proteins, be it receptors, be it like actual cells, be it causing instability in the systems. I think that the easiest one to highlight and the one that I spent the most time on for some reason, because I thought it was like really difficult to collect, is the blocking of neurotransmission. So the example that the paper talks about is the same example that Anna used in class, which is MG. I don't want to try to pronounce the name of that. I think it's myasthenia, but I could be butchering it. Myasthenia gravis. And so basically what they have. What?
I did that topic too. It was me and Anna.
My bad.
Was it just Anna?
Sorry, it was Anna and Neha. My bad. My bad. You too. And so what ends up happening, that if you read page 10 of that paper and you understand it, let me know how you understood it, because it took me a while. But basically, acetylcholine is used as a receptor, and if you want to do a bit on it and talk about it too, you can. I might force you to, but so if there is the usage of acetylcholine as a neurotransmitter, as a molecule. And the autoantibodies tend to be reactive to the receptor molecule for acetylcholine, correct? And so what ends up happening is there's a prevention of the continuation of the neurotransmission because of the blockade of the autoantibodies. And then the antibodies, from what I perceived trying to read the paper, because it's super dense, the image like makes it a little easier. It's almost like a phagocytosis process that happens with the receptor once it's bound to an antigen for some reason. I don't know, like I didn't understand why the process happened. But not only you're preventing the receptors from working, you're also like just straight up inducing the receptor to go back into the cell. And I didn't really understand why the process happened. But yeah, so I just thought that out of like those 7 examples, that one was like the easiest visually to understand. But there's highlights on all 7 of those in that paper. If you can read like the little monologue before they actually start talking about the process in the paper, that's also helpful. And yeah, and then just the last thing that we needed to cover was autoimmunity versus autoimmune diseases. So autoimmunity means that there exists the autoreactivity of antigen binding domains and T and B cells. Okay, and this happens because there are just too many endogenous particles and too many exogenous particles that are pretty similar. And so if you were to fully remove the ability of the immunoglobulins and the TCRs to react to more compounds, by default, you would hinder adaptive immunity. And so the existence of autoimmunity and like autoreactive antibodies and TCRs is just something that the body accepts as a means to further protect you from adaptive or from pathogens that it should recognize. And so it's just basically like taking the hit and accepting the fact that it can have autoimmunity in order to protect you further from a larger number of pathogens. And the biggest difference is that autoimmunity can be non-harmful, while all autoimmune diseases are harmful. And the initial part of the paper, like if you read just the abstract for that 42-page paper or whatever, says that like about like a lot of healthy adults have autoimmunity going on in their bodies, but only about 2.5% of the population has some sort of autoimmune disease. And a big thing is that autoimmune diseases are caused by autoimmunity, but not everybody that has autoimmunity will have an autoimmune disease. Yeah. So thanks for coming. We hope this was helpful. There's a lot of content on this material, which, you know, can be very challenging, but you can feel free to reach us with questions. And yeah, we hope to see you here next week to talk about microbiome. Cool.